Hello, I'm Boutania Ettetuani, PhD student from Morocco. I'm here to present you the role of the gene ontology article after Microarray Gene Selection. Our approach, as you can see in the figure number 1, has aimed to identify significant differential expressed novel and common gene and other variants from various processes, then a co-expressed gene based on the similarity between genes, not only based on their expression but also focusing on their association to construct an ontological analysis which study a set of glomerular diseases case in parallel rather than an individual term. Exactly when measuring the distance and before evaluating the clustering analysis algorithm. After all, an hypothesis evaluation and validation. Our data focus on the transcriptome analysis of human diseases, which the glomerular diseases include many conditions with a variety genetic and environmental cause. For the method, the statistical method used to detect the difference expressed gene, as you can see in the figure number 2, was the parametric method within the t-test, which fault change was 2 and the p-value of 0.01 were estimated as a prior probability that gene is differently expressed. A total of 82 genes were clustered to find and define the best similarity value and with a minimum of negative value between a gene based on the gene ontology term on all three aspects: molecular function, cellular component, and biological processes by applying the Wang method to calculate similarity. 6 cluster was found, as you can see in the figure number 2, presented as a hierarchical clustering for the distance measure. For the hypothesis evaluation, all co-expressed genes were used to search for shared gene ontology function. This kind of analysis generates a huge quantity of initial results containing very general terms which need to be given a biological interpretation. Gene ontology enrichment analysis difference between each cluster. The top we, as you can see in the figure number 4 and the figure number 5, we input a gene from literature and our difference expressed gene to compare it and to give an interpretation of the result. The top pathway can be displayed as seen in a plot, as you can see in the figure number 4, simplifying the result and displaying all enriched terms with the enriched score visualizing the linkage of the gene and the biological process. The second form of the hypothesis validation was focused on the investigation of the molecular underpinning of specific human diseases. A heat map plot was used to visualize the association of co-expressed genes to the disease. The color of the heat map is rated the score of each association, so the darkness blue are that's one who have a high score of association. For the conclusion, the data analyzed here focus on the collective cluster of different expressed genes from patient undergoing the diagnostic evaluation. And the next step for the analysis is to suggest a new function that are not reported in the literature study for the detection of the novel gene based on the score of expression and the score of gene ontology in parallel. And thank you.